the Zoomy stuff?
Except the full view. Go back to this one.
Oh, there we go. There we go. All right, everybody, can you see the slide set?
Yes, we can.
Fantastic, okay. So I'm gonna change it up just a tiny little bit today. So this is a session that we were gonna do later on in the course, but I figured it'd probably be good just to go ahead and maybe get this one out of the way so it can help kind of support some of the future discussions that we'll have over the next 2 days. So we talked a little bit yesterday about DNA, just kind of some basic stuff about some of the characteristics of the DNA molecule. We talked a little bit about base pairing, and then we spent some time talking from a forensic perspective on some techniques that are used to examine DNA samples. We talked a little bit about the RFLPs, the goods and the bads of that. We talked very briefly about some sequencing stuff. We talked about paternity and maternity and how these, when you do restriction digestions with DNA molecules and you cut them and then you run them on the gel, on the scientific jello, you can see different banding patterns that you can use to kind of compare back and forth. So from a very 10,000 foot view, we talked a little bit about genomics. And so some of the upcoming sessions will build on the stuff that we talked about yesterday. And so today I want to spend a little time talking about immunity in the immune system. And this will be a brief overview of the immune system in general. And again, I'm doing this so we can make sure that when we talk tomorrow and Thursday on the other sessions, that we have some background in what, some basic characteristics of the immune system are. So we're going to spend some time today talking about the immune system. This is something that you don't see a lot of people take anymore, partially because these classes aren't offered as much anymore as they used to be. Certainly at the graduate level and above, particularly at medical school, or if you go to a doctoral program, most places are going to offer immunology at some level. I used to teach undergraduate immunology in the College of Science many years ago down here. But again, it's not something that's taught very much today. However, a lot of us, if we're going to get a science degree and we're going to go into research, or we're going to go into a career, maybe working at a biotech company or something like that, most of the time we're going to be focused on something that's going to be treating a given disease. We're going to be focused on maybe designing a new drug that can help treat HIV infected individuals, or we want to help design maybe a new vaccine, or we want to try to design a biomarker molecule. So maybe you are interested in studying cancer cells, and you know that cancer cells express a very high level of a certain biomarker than other cells do. So if you could maybe figure out a way to tag those cancer cells by targeting that biomolecule that has a very high level of expression, then you can differentiate between cancerous cells and non-cancerous cells when you look at them under a microscope, or if you do some other testing. And so a lot of this comes back to the immune system and having a basic understanding of how the immune system works. And when you understand how the immune system works in a normal, healthy individual, then when you see somebody that's sick or diseased, you have a better idea as to how to treat them or what may be going on with them. And from a research perspective, we use a lot of the components of the immune system to help drive drug discovery, to test out new theories for disease immunity or how diseases and infections progress. So it's always a very good idea to have some kind of overview of the immune system at some level. So this is going to be kind of a bastardization of an immune lecture that I give to medical students, which is generally kind of at about the upperclassmen, beginning graduate student level. So I won't get too far into the weeds today, but we're going to go ahead and cover the immune system. And again, just as we did yesterday, I'll do my best to try to catch people if you put a hand up or something in the Zoom. But if you've got a question, it's probably best just to unmute yourself and just say something, so we can stop the bus and address your topic. So let's talk a little bit about the immune system. So what is the immune system in general? And this is something that probably many of y'all have heard before. Probably not too much new is going to be new here, but the immune system is essentially a system within most organisms. And even if we look at insects, we look at plants, we look at animals, all of these organisms have immune systems. Now, the immune system in a mosquito is going to be different than the immune system that you and I have. But many biological organisms, particularly many of the higher organisms on our planet, have got an immune system, which is basically a system within the organism that allows for you to be able to identify and eliminate pathogens. So I've got some definitions down here in the bottom. When we say the word pathogen, what does that mean? For the purpose of this lecture, it's anything that can cause a disease. This could be a virus, a bacteria, fungal infection. It could be parasitic worms. It could be a mold, etc. So anything like that we'll refer to as a pathogen. An antigen is essentially a substance that is able to elicit an immune response. So if you walk outside the last few days, it's been windy as hell down here. We got a little bit of rain. It's spring. And so a lot of the flowering plants are going crazy. There's a lot of pollen in the air. Everyone's having allergies. I know my kids right now in particular are suffering with allergies. And those allergies are caused by the pollen grains getting into your body. When you walk outside, you take a breath, you get some pollen in your system, and this is going to then activate an immune response. So the pollen would be considered an antigen. If you are running down at the beach at South Padre and you step on an old rusty can, that rust that's on the edge of the can and would be considered an antigen if it's going to get inside of your system, and it would elicit an immune response. If you poke yourself with a splinter, the little piece of wood that gets stuck in your skin is going to elicit an immune response. And so you could technically consider that an antigen. So an antigen is going to be anything that elicits an immune response. And we'll talk about how an immune response is elicited here in the next couple slides. As well as having antigens that elicit immune responses, we also have a special class of cells within the body that we refer to as APCs, or antigen-presenting cells. And these are cells that play a very specific role in the immune system, and their job is to basically roam around your body looking for foreign substances, and then once these APCs encounter a foreign substance, they will consume it through phagocytosis, and then they take that foreign substance that they've encountered, they digest it, and then they take a tiny piece of that digested substance and shove it out to the surface of the cell to display it to other cells of the immune system. So this is very analogous to thinking about, if you've got a Roomba or you have one of these little robot vacuums at home, it'll run around your house gathering everything up. The difference between the APC and the Roomba is that after the APC has gobbled up everything that it found in your house, it will then show you what it found. Hopefully your robot vacuum doesn't come to you and show you everything that it picked up. But we have cells in the body whose job is to go around, gobble up things that are floating around, and then show them to the other cells of the immune system, so the immune system can assess that substance and try to figure out, is this good or bad? Is this something that I need to activate an immune response to, or is this a normal protein that's not going to cause me any harm? And this leads to the last term, which is presentation, which is how these antigen-presenting cells will show or present the foreign substance, the antigen that they've encountered, to the cells of the immune system. So we're going to talk about today in this lecture the primary features of the immune system. We'll distinguish between what innate and adaptive immunity is, and then we'll talk about a little bit about B cells and T cells, how they're formed, what their basic roles are, how long they persist in the body, and what their utility is within the context of having an immune response. So some general features of the immune system. As I said a couple slides ago, it's probably the oldest and most widespread system that's shared amongst all the multicellular life forms on Earth. If we look at animals that have been around for millions of years, sharks, whales, many marine mammals, they have got immune systems. We can actually track some of the lineage of our immune system characteristics back to these animals. And so the immune system has probably been around for just about as long as there's been multicellular life on Earth, which would kind of make sense if you think about it from an evolutionary perspective. If you are an organism that's evolving in a new environment, you probably need a way to protect yourself from that environment. Now, that could be something like claws or teeth or wings. Maybe you can run really fast to escape predators. But what about the predators that you can't escape? Viruses and bacteria infect animals just as much as they infect human beings. And so these creatures also need a way to be able to fight off infection. So The immune system is very ancient, very widespread. We see it in virtually every multicellular life form that's on the planet that's still around today. The immune system is a complex system that's very distributed throughout the body that's self-renewing. So this would be very much analogous to you having a car that you never have to fix, because if the car starts to break down, it can automatically repair the brakes. It can automatically repair the gasket in your engine. So our immune systems are able to basically renew and repair themselves all the time. When you get sick, you get an infection, and you have a dramatic increase in the number of immune cells in your body to fight the infection. When the infection goes away, the numbers of the immune cells will come down. They won't go away, but they'll come down, and they'll be ready to, and they'll renew themselves. They'll make more B cells and T cells, so they're ready to respond to the next system, to the next infection. So some key hallmarks of the immunity and the immune system in general is that it's very adaptive. The immune system is able to train itself to react to new threats. And we'll talk a little bit about how this happens in a moment. So this means that if you think about, we've talked about sending people to other planets. We've already sent people to the moon. We're talking now about, colonizing Mars. That's Elon Musk's big deal, is Occupy Mars. So if you are a new astronaut going to Mars and you encounter a Martian bacteria, how would you be able to survive that bacteria if you've never seen it before? Well, your immune system has the ability to train itself to react to new threats. And we'll talk about how this goes in a moment. So the immune system is very adaptive. Assuming that you can survive the infection long enough, your immune system should be able to eliminate the infection once it has a chance to train itself how to respond. The immune system is very error tolerant. A mistake is not fatal, which, if you think about this again from the perspective of the whole organism, we have about 6 billion cells in our body. About half of those cells turn over every day. So every 24 hours, you're having about half the cells in your body being destroyed and then regenerating themselves. That's 3 billion replication events. We talked yesterday about DNA replication, and how DNA replication, you usually have, for every million bases that you replicate, you have about 1 to maybe 2 errors. So if you're talking about replicating an entire DNA molecule, which is about 3 billion base pairs for a human being in one cell, and you're going to have 3 billion cells replicating themselves every day, there's a lot of chances for mistakes to occur within the context of DNA replication, but the body has developed a system to be able to address these mistakes if they occur. The immune system has also developed a system to be able to tolerate mistakes. And so if you accidentally produce a T cell, for example, that decides that it wants to react against you, this is what forms the basis for autoimmune diseases like lupus and muscular dystrophy and other things like cystic fibrosis, where we have an arthritis, rheumatoid arthritis, where you have the body attacking itself. In order for this to not be a fatal mistake, the immune system has to have some tolerance for errors. And so this tolerance for errors is what leads to the immune system being a benefit for us. But at the same time, if a mistake is made, you're not going to wake up dead in 5 minutes because your immune system made a mistake. The immune system is also self-protecting. As we've discussed here already, it's able to protect itself. If it sees something new, it can develop a modality to be able to respond to this new pathogen and self-protect. And lastly, the immune system has the ability to remember an encounter with a pathogen or an antigen, and this can allow it to respond quickly if it encounters that pathogen again. We refer to this as immunological memory, and memory forms the basis for what we do when we make a vaccine. You make a vaccine and you introduce the vaccine to somebody. So we've all, by the time we're 1 or 2 years old, we've all had generally half a dozen vaccines. When you're a baby, you're going to get vaccinated against the DDT vaccine. You're going to get vaccinated against diphtheria, tetanus. You're going to get vaccinated against measles, mumps, rubella. For many people, this is a one-time deal. You'll get these vaccines when you're a baby or a child, and then you'll never get vaccinated against measles again, but you'll have lifelong protection from measles. Well, how is that possible? Because when the immune system encounters a pathogen for the first time, once it clears the infection, some of the cells that were used to clear and fight that infection are basically saved. They're saved in a special hard drive, if you will, within the body, a special location within the body, and these cells become memory cells. So if you happen to encounter measles again, your body's able to immediately generate an immune response, and this protects you from getting infected. And this basis of memory is what's formed the foundation for modern vaccine development. I don't count the COVID, the SARS vaccine as a vaccine because that thing's not a vaccine. It's a drug, it's a treatment. Nobody that took the COVID vaccine was immune from getting COVID after they took it. I don't know how many of y'all got vaccinated, but I did and ended up getting COVID a couple times after the vaccine anyway. I've been vaccinated against measles, yellow fever, smallpox, anthrax. I've never been infected with those things after I got vaccinated against smallpox or against measles. And we have measles outbreaks, sadly, in America quite a bit because we have a subset of people within this country that believe that all vaccines are bad. And so they don't give their children vaccines. And then these kids go to school, encounter maybe another kid that's got a disease like measles, and then they get an infection. We've had several big measles outbreaks in the last 15 years, primarily in California among richer parts of the population because wealthy people have gotten this idea, particularly wealthy white people, that vaccines are very bad. And so they don't vaccinate their kids. If you send a kid to school without a vaccine, you're really, ensuring that that kid's going to get sick at some point. So the immune system's ability to remember encounters and then save that memory so it can respond quickly upon, reinfection is a very critical component of the immune system. Probably one of the most important of these hallmarks right there amongst the four is the ability to remember an encounter. So we have this system, very old system. It's very robust. It can remember things. It can respond to virtually every pathogen that it comes across. So what are some of the advantages of the immune system? And we're going to kind of talk about this talk here in general. One of the advantages is that this is something that is pretty much standardized amongst all of our species. So if you learn the immune system and you study it in patient A, patient B is going to have the exact same immune system. Now, are there exceptions to this rule? There obviously are. We have some people that have mutations in some of the cells of their immune system where they don't respond quite as well. But by and large, if you learn the immune system and how it functions for one person, this is going to be applicable across many, many people. And so in the talk today, as I said earlier, I'm going to try to keep this as simple as possible. We're going to really focus on just the basic mechanisms that you need for defense and adaptability, and try to make sure that this is something that people can kind of understand and take away with some knowledge from this lecture. So let's talk about the first line of defense in the immune system. So there's really two lines of defense within the immune system. We refer to these lines of defense as innate and adaptive immunity. So we're going to talk a little bit about innate immunity first. So the innate immune system, the innate component of the immune system is basically a series of barriers that are non-specifically designed to respond to pathogens. Well, what do I mean by this? This means that these are a series of barriers that are able to protect you against all types of pathogens, whether they're viral, bacterial, fungal, parasitic worms. But we have a basic set of systems within the body that comprise this innate immune response. And these systems are primarily going to be physical systems. So if you look at the graph or the table that we have over here, on the top, we've got skin, the gut, your lungs, and then your eyes, your nose, and the oral cavity, which of course would be your mouth. This could also be the anus as well. And then over here going down on the left side, we've got mechanical, chemical, and microbiological. So the way to read this table is let's start up here with the skin. If we look at the skin and look at some of the characteristics of the skin, if you look at your skin right now, hopefully your skin is intact. You don't have any cuts or scratches. And those cells that make up your skin are epithelial cells that are very tightly connected together. And then over the layer of our skin cells, we have a very light coating of wax that's keratin. And so we have this waxy covering of cells that are very tightly joined together, so tightly joined together that it essentially makes our skin waterproof so long as our skin is intact. And this very tight layer helps to protect us from pathogens. If you think about what's under your skin, it's going to be your blood vessels, some additional tissues. Those substances are not very resistant to pathogens. That's where the pathogen wants to get. So if you have an intact layer of skin, then this is going to be very, very effective at protecting you against pathogens. This is why we often tell people when they work in high containment facilities or they work in research labs, check your hands to see if you have cuts. If you have cuts on your hands, then depending on where you're working, it's probably a good idea to not go into work until that cut is healed, or to make sure that it's covered up with a Band-Aid or something like that, because an open cut on your skin gives pathogens access to the structures that are beneath your skin. But if your skin is intact, it's going to be a very, very good way to protect you from pathogens. If your skin is intact and the waxy layer is intact, then every time air blows over your skin, when you're walking around, you're in a room, that airflow is going to help constantly push pathogens away from you. At the risk of freaking everybody out, we have microbes falling on us all the time. If you look up at the ceiling right now and open your mouth, you can probably eat 5 or 10 million bacteria. We can't see them, they're very, very small, but we have crap falling on us all the time, whether we're outside, inside. There are things that we can't see that we are encountering on our skin all the time. So if the skin is intact, these things are going to basically blow right off of us and just kind of float away. In addition to that, we have chemicals that make up our skin, such as fatty acids, and we have a general class of defensive proteins that we refer to as the defensins that are there to help clear away any pathogen that might be able to penetrate the skin. So we have and they're going to respond to any kind of pathogen, bacteria, viral, a parasitic worm, pollen from a plant. So we have a very non-specific way with our skin to be able to provide us protection that's not focused on only protecting against a virus or a bacterium, but it's going to provide us protection against many different types of pathogens. And as you go across the table, you see the same thing. So if we look at the gut, and many of y'all probably know this, inside of our stomachs, we have a very low pH. Our stomach contents are very acidic. This is going to help break down a lot of pathogens that we might consume. So when you go maybe eat those tacos from the street vendor that have been there for 2 days, maybe the meat or something is starting to go bad a little bit, but the low pH in your stomach, assuming that you don't eat too much of them, should help you protect you against infection. We also have enzymes in our stomach that are going to work to help break down not only the foods that we consume, but any bacteria or viruses that may be consumed along with those. And then again, we have another general range of these defensin proteins that hang out in our stomach that are able to respond to any kind of bacterial or viral infection that we may end up ingesting. So we have a very non-specific way in our stomach to treat these things. The same thing with the lungs. Everyone knows that our lungs have cilia. We have these tiny little hair-like projections that are always moving inside of our lungs. When you cough, it's generally because cilia have started to trap dirt and other substances that we breathe in. And so the cilia will start to wave back and forth. This stimulates the muscle cells that the cilia are attached to start to reflex, and this causes coughing. And then when you cough, you're helping to push whatever you might have inadvertently breathed into your lungs back out. The cilia are coated with mucus. They're coated with mucus. And so this is going to then help trap any viruses or bacteria on the surface of the cilia so they cannot reach the cells underneath the cilia. And again, when you cough or when you cough up a booger or something and spit it out, you're going to be spitting out these pathogens. We also have surfactant, a chemical within our lungs that's used to help keep the water in our lungs from sticking to the lung tissue so our lungs don't collapse. But there's another benefit that we get from surfactant, which is because it's helping to keep the water away from the surface of the lungs as much as possible from the cells. This is also going to help keep pathogens away from the lungs, the cells in the lungs as well. And then just as we saw for the skin and the gut, we have a general class of defensin proteins that are in our lungs that are there to help protect. And the same thing with your eyes and your nose and the oral cavity. We have chemicals and defensive proteins in these areas that can help fight off infections. And then within all of these areas, the skin, the gut, the lungs, and our eyes, we have our own bacteria that are growing there, that are supposed to be there. Bacteria are very tribal. They don't like other bacteria coming into their hood and trying to drop, stop, and set up shop. They want to keep those bacteria away. So the bacteria that live in your stomach, for example, if you accidentally consume something that's infected with bacteria, the stomach bacteria are not going to want those things to live in the stomach with them. And so they're going to fight against them to kill them. We have bacteria that live on the surface of our skin all the time. We have staph bacteria living on the surface of our skin. Again, the staph bacteria is supposed to be there, but it doesn't want other bacteria to live on our skin. And if it encounters other bacteria, the staph bacteria is going to secrete antibiotics. This is where antibiotics come from, to kill the other bacteria. So the normal bacteria that live in our stomachs, on our skin, and in our lungs are going to help keep us protected from other pathogens. And so the innate arm of the immune system is basically a general way of fighting against pathogens without specifically targeting a certain pathogen. We have a separate arm of the immune system that's able to specifically target certain types of pathogens. So if we look at this flow chart here and just kind of walk through this, you're the pathogen up here. You come in and you get exposed to a new host, and that new host has got an intact skin barrier. Let's say you are an influenza virus and you just got coughed out of somebody's mouth and you're floating around in the DMV and it lands on your arm. Well, if you've got an intact layer of skin where that thing has landed, then the infection is going to be prevented. Let's say that influenza virus that was coughed on you at the DMV is able to get through a cut in your skin. Well, then you're going to have the beginning of the infection. But when the infection begins, since it's on your skin, you're going to have a non-specific response from the innate immune system, and some of those bacteria that live on your skin, some of those defensive proteins that are present on your skin are going to start fighting against that pathogen, and more than likely they will defeat the pathogen and remove it from the skin. If that doesn't happen and the pathogen's able to establish infection, then what are you going to do? This is where we see the next arm of the immune system come in, which is the adaptive immune response. And this is the immune response that most of us know about, that has B cells, T cells, antibodies. This is the part of the immune response that's going to be very specific against a very specific pathogen. So let's move on and kind of look at this a little bit more. So let's say you are a brand new unseen pathogen. You are Elon Musk's number 2 person you're the second person out of the rocket ship on Mars. And you walk around on Mars a little bit and you encounter a brand new bacteria. Somehow it's gotten through your filtration system in your suit, and you've breathed this bacteria in, and this bacteria has been able to evade the immune response. You know, if you're a pathogen coming in, you're like, hey, I just defeated half the immune system. Now all I got to do is get in here and replicate for a little bit and make more copies of myself, make a few million friends, and then I'll be able to establish an infection. But if you're a brand new pathogen that the immune system has never seen before, how is it going to be able to get rid of you? If you've already gotten past all the barriers that were designed to clear non-specific pathogens and you're brand new, you've never encountered this human being before, how is this human going to be able to clear you and get rid of you? And this is where the adaptive immune system comes in. So in addition to having this non-specific system that can fight against pathogens, we have a system of cells that make up the immune system that have the ability to very specifically target certain pathogens and eliminate them completely from the organism. And what we're basically talking about here are B cells and T cells. These all come from a general class of cells that come from the blood, that come from the bone marrow, that are called lymphocytes. And we'll talk more about that here in just a moment. But what we have is these general cells that are called lymphocytes, and these lymphocytes are going to be the cells that are going to respond to the infection. Lymphocytes are primarily going to be either B cells, which secrete antibodies. B cell makes antibodies, or these are going to be T cells, which have the ability to directly kill an infected cell. And so it's going to be the innate and the adaptive immune system that work together to eliminate pathogens while also preventing the organism from being destroyed from the immune system. So we're sending out T cells to go out and kill infected cells within the body. We need to have a way to control those T cells, because if the T cells just start to go crazy and kill every cell in the body, that's going to be very bad for us. And so we need to have a mechanism by which the T cells and the B cells are very tightly controlled. So when they start to respond to an infection, they don't start to kill healthy cells in that area. They only go after the cells that are infected or that are sick. So these B cells and T cells are going to be responsible for producing antibodies and for directly killing infected cells. And so we refer to the adaptive immune response, since we have B cells and T cells, it can really be subcategorized into two categories. Humoral immunity, which is the production of B cells, and cell-mediated immunity, which is T cells killing infected cells. And so now let's kind of walk through that a little bit here. So I talked a little bit a few slides ago about antigen presentation, and this is just a concept. It's a very important concept within the immune system, which is essentially, as I spoke about a few minutes ago, we have cells in the body that are running around and they're eating everything that they find. They're very hungry. They're hungry hippos, right? They're going around, and these cells, we refer to these cells as phagocytic cells because they use phagocytosis. Phagocytosis, for those of you that might not remember, is the process where cells eat. The cell can basically it can basically wrap projections of the cell. This cell rearranges its cytoskeleton inside of it, and it can form these little arms, we call them pseudopods, that will engulf a foreign substance. It could be a substance from you. It could be a piece of protein from you. But anything that these phagocytic cells find floating around outside in the body, they will eat. Once they eat these things, they're going to digest them. Once they've been digested, they take parts of whatever they've eaten and they push them out to the surface of their body, and then they show them to other cells of the immune system. So these other cells of the immune system, such as the T cell in this cartoon, it looks at what this antigen presenting cell, or APC, has consumed and what is it presenting, and then the T cell looks at it and says, hey, what is this thing? Is this foreign? Is this something from outside of the body? Or is this me? Is this from the body? The T cell is able to delineate between self-peptides or self-antigen and foreign antigen. And this concept of presentation is really the foundation for the adaptive immune response, because if the T cell looks at what's being presented to it and it determines that that thing is danger, it will then activate an immune response against that specific pathogen. So if this phagocytic cell has eaten a piece of an anthrax bacterium, and it digests the anthrax bacterium and shows a piece of the anthrax bacterium to this T cell, then this T cell is going to say, hey, everybody, we have anthrax. Everybody that can fight anthrax, get over here and start fighting anthrax. And cells that are able to fight anthrax, and only anthrax, will start to respond to that area. This is what gives the immune system, it's the adaptive immune system, what we call specificity. The ability to specifically target a pathogen. And this is what keeps the T cell from randomly killing every other cell in the area, because the T cell can then go and check every cell in the area where the infection is and say, hey, are you sick or are you healthy? This is like the cops coming to your door and knocking on the door to do a check to make sure you're okay. You open the door and say, yeah, everything's fine. then the cops leave. If you don't answer the door and they hear screams coming from inside the apartment, they're probably kicking the door down to come see what's going on. The T cell is doing the same thing. If it checks the APC and it finds out that the antigen is not harmful, maybe it's a piece of your stomach protein or something else. It's a piece of a cell that died that's from you. It's not going to generate an immune response. But if the T cell determines that, hey, this is a piece of anthrax bacteria, this is a piece of the influenza virus, then it's going to activate an immune response against that pathogen. Does everyone understand this concept of antigen presentation? Is that clear? Okay. So now let's talk about how T cells are formed. So all of these cells in general, I think I may have had a slide in here, I don't. Okay, so all of the cells of the immune system come from your bone marrow. So this is why when people get certain types of cancer, they maybe get leukemia or they get certain types of bone cancer, we will do a bone marrow transfer where we will basically try to find people, usually people that are related to that individual, that will match up with them genetically, and then we will basically give them a very super high dose of radiation, like a Bruce Banner dose of radiation, and this will kill off all of their bone marrow, and then we will give them new bone marrow from a donor. The idea behind this concept is that that new bone marrow from the donor is coming in from somebody that's healthy, that's a genetic match for this person, and then the new bone marrow is going to begin to grow. It'll spread out to all the other bones in the body. And then from this new bone marrow, new cells are created. Some of the new cells that are created from the bone marrow are immune cells. So when people get certain types of cancer, this is kind of the concept behind doing a bone marrow transfer. Because all of these cells within the body, many cells within the body, come from the bone marrow. And the immune cells are no different. So in the bone marrow, you're going to have this mama cell, for lack of a better word. It's called a pluripotent stem cell, but you have this mother cell come out of the bone marrow. And once it comes out of the bone marrow, it's going to start to get exposed to different chemical environments within the body. And depending on what those chemicals are, that mother cell will then start to turn into either a B cell or a T cell. And then these cells will begin to spread throughout the body. And this is a process that's happening probably tens of billions of times a day. Because remember, one of the characteristics of the immune system is that it's self-renewing. And so you have all these cells coming out of the bone marrow. They're getting, maybe some of them end up in the spleen, maybe some of them end up in the thymus. They get spread around your body. And depending on what the chemical environment is in their new neighborhood, is what those mother cells are going to turn into. They're going to differentiate into. So we have T cells that come out of these mother cells that come out of the bone marrow, and they turn into, we're just going to call them baby T cells for lack of a better term. When these baby T cells come out, they have to pass two tests. The first test is, can these baby T cells bind to MHC? MHC is the protein that antigen presenting cells use when they grab antigen and show it to everybody else. So if we back up to this cartoon right here, in this cartoon where the APC is presenting antigen to this T cell, it does this with a molecule called MHC. So MHC basically grabs this little piece of whatever this antigen presenting cell has eaten, and they come together and then get shoved out to the surface of the APC for recognition. So the T cell test number one is, can this baby T cell recognize and bind to this MHC molecule? This is test number one. All of the T cells that are able to bind to MHC pass the first test, and they get put over here in this corner. If you are a T cell that comes out and you cannot bind to MHC, then you're not going to be very useful, and then you get put in another corner and you get recycled. So the first thing with T cells that the body checks is this cell going to be functional? Can it recognize this MHC molecule that's going to be holding antigen from all these other antigen presenting cells? Yes or no. If it's a yes, then you pass the test and you get to go to the second grade. If not, you get recycled back to kindergarten. That's the first test. The second test that the T cell has to go through is, okay, you could bind to this MHC molecule. That's great. How tightly do you bind? Do you bind so tightly that you never let go? Or do you bind tightly enough to where you hold the T cell next to the antigen presenting cell so you can signal and do some things, and then once you're all done, you release and let go. So the second test for the T cell is how tightly are you going to hold on to MHC? If the T cell holds, this is kind of the Goldilocks test. If the T cell holds on too tightly or not tightly enough, it gets put in the corner to be recycled. If it holds on just right, just perfectly, just tightly enough to where the cells can be held next to each other so they can signal, but not so tightly that when the signaling event is done, the two cells can break apart. If the T cell can pass that test, then it is considered to be a mature T cell, which is right here. And now it's allowed to go through the body and start to fulfill its functional role. So in normal individuals, this process is called selection. This is called positive and negative selection. The positive part is, can the T cell bind to the MHC molecule? The negative part is, can it let go? So can it bind over here and then can it let go when it needs to let go? If those two things are a yes, then we save that T cell and it becomes a mature T cell. If either one of these are a no, then that T cell gets recycled. This is what forms the foundation for autoimmune diseases. Sometimes, and again, this process in a perfect world would be perfect, but because we're not perfect organisms, we do have instances where T cells will basically dodge these two tests and they will escape this process of selection and they'll get released into the body. And so in what we see in people that have things like lupus and cystic fibrosis is that their T cells are binding too tightly to the self MHC molecules and they won't let go. They're not supposed to be binding there really at all, but then they bind and they stick and they will not let go. And when cells are stuck together and they're connected like this, they're going to be signaling to each other. These signaling events are going to cause these cells to release certain types of chemicals that are going to start to affect other cells in the body, not just immune cells, but all the cells in that area. And so this is what we think leads to autoimmune disease. And we think this because we've been able to use mice that we have genetically modified their immune systems to where they don't make T cells or they don't make B cells. And then we infect them or we do certain experiments with them to replicate lupus or other disease conditions. And we see the T cells responding in these manners. And so, at least as far as we understand right now, because T cells sometimes don't go through the selection process very well, and they get released into the body anyway, this forms the foundation for autoimmune diseases. And so T cells are very, very important in terms of them being able to bind to infected cells and then release that infected cell once the T cell has done its job. So what is the T cell's job? Once it gets made, it's mature, it's floating around out there. What is the T cell's job? The T cell basically is going to have two main functions that it's going to perform, and it's done by two different types of T cells. I don't want to get too deep in the weeds here, but when you have a mature T cell, this mature T cell can take one of two roles. It can be a killer T cell, a cytotoxic T cell. We refer to this as a CTL. This is the T cell that just goes around the body, knocking on the door, saying, are you sick or are you healthy? If you're sick, I'm killing you. If you're healthy, I'm leaving you alone. That's all this cytotoxic T cell does. It floats around the body, knocking on every door, checking every cell. Are you healthy or are you sick? If it's healthy, the T cell leaves. If it's sick, the T cell basically sends it a text message that says, hey, you should unalive yourself. And the cell goes through apoptosis and the cell kills itself. So under normal conditions, you're going to have some T cells roaming around the body, checking on all the cells, saying, are you healthy or are you sick? We have another category of T cells that they roam around the body, and their job is to basically look at all the antigens that are being presented and then start to secrete chemicals if they decide that the antigen is a pathogen. So we have these antigen presenting cells, they're gobbling up all these things. They show it to some T cells, and some T cells say, man, we got to go find every cell that's carrying this antigen and kill it. And some other T cells look at it and say, hey, this is bad news. We need to tell everybody, hey, we have an infection with anthrax, and start to signal and tell everybody. So the T cells play a very, very critical role in being able to kill infected cells and also being able to signal and tell other cells of the immune system, as well as other cells in the body, hey, we are sick over here in the stomach. So we need help responding to this infection in the stomach. And then T cells are able to, as the cartoon shows down here, they migrate around all throughout the body looking for infections. There is one area in the body where the immune system is not allowed to go. One area, actually two, I take that back. The one area is your brain. So if you think about every time you've gotten an allergic response, you walk outside right now and there's pollen blowing everywhere and you start to sneeze and cough, you're having an immune response to the pathogens, to the allergens. If you had that kind of response in your brain where you have T cells running around and killing brain cells, that's probably a bad thing, because there's one cell type in the body we have a really hard time making new ones of, and those are brain cells. Once you lose them, they're gone. If you start to have an immune response against the cells in your brain, that's a bad thing. And so there is a physical barrier called the blood-brain barrier that basically surrounds your brain and protects your brain from the immune system. And there's also essentially a wall, if you will, in your central nervous system that says no immune cells. And so your brain and your eyes do not have any immune protection because the immune system is not allowed to go there. And again, if you think about the job that some T cells perform, where they go around and kill infected cells, what if that T cell makes a mistake, which sometimes happens, and it says, oh, the cells in your eye look like they're infected, I'm going to start killing them. Nobody wants to go blind because your T cells are killing your eyes. So the immune system does not allow, the body rather, does not allow the immune system to go into your brain. The other area in your body where the immune system does not allow a response to occur are your sex cells. So for men, these are going to be the Sertoli, the cells that make sperm. And for women, these are going to be the follicles in the ovary. So the cells that we use for reproduction, primarily the sperm, the cells that make sperm and the cells that make the eggs, they do not get checked by the immune system, nor do your brain, nor does your head, nor does your brain or your eyes. Everything else is fair game. And so T cells are going to be floating all around your body, constantly looking for cells that need to be killed. Sometimes cells need to die, not because they're sick, but just because they're really old and they're not functioning as well. And the T cell's job is to go basically, like I said, to use a bad analogy, it's like the cops coming to your door and they're knocking on the door. Are you okay in here? Yes or no? Okay, you're okay. We're going on to the next house. Only in this example, the T cell knocks on the door. Not only does it ask if you're okay, but it asks, how old are you? And if you take 20 minutes to answer the door because you're 200 years old and you can barely get up off the couch, the T cell is probably going to say, okay, it's time for you to recycle yourself. So T cells play a very, very important role in the immune system. And again, we've done studies in mice where we've taken out the mice's T cells, and those mice do not live very long. We have seen in our own DNA studies in humans that if you have a mutation in your T cells, when you are a fetus and the T cells don't form, that's a lethal mutation, and that fetus is not going to make it to term. So there are some mechanisms in the body where during fetal development, the fetus is checked. And if the fetus doesn't have the ability to do some of these things, that's generally considered a lethal mutation, and that fetus is not going to make it to term. This is a check that happens early in development. You're not going to have somebody who's 3 or 4 months along suddenly lose their baby because their immune system doesn't work. The baby would never get that far. This is a check that probably happens within the first 10 to 14 days following conception. But mutations in the immune system are very, very serious because we need the immune system to play such a vital role. So how does the T cell do a lot of its job? It does it with a molecule that's called the T cell receptor, or the TCR. And this is going to be the molecule that the T cell is going to use to interact with this antigen presenting cell. So these are all T cell receptors that we see on this little purple cartoony cell. And this T cell receptor is critical in giving the T cell its ability to respond. This also is how our immune system is able to fight new pathogens, even if it's something we've never seen before. Because this T cell receptor, also known as the TCR, can go through, it has several different proteins that make up the T cell receptor. Here's the alpha chain, this is the beta chain, and then it has these accessory molecules that are over here. This is the T cell receptor right here, and this accessory molecule. These things all need to function properly for the T cell to be able to bind. And as many of y'all probably remember from basic cell biology, when receptors interact with other receptors, it's kind of like a lock and a key. There's generally only going to be one way for this fit to occur. The T cell is able to get around that limitation by using a mechanism that we call VDJ recombination, or junctional diversity. These are all fancy scientific terms to be able to say, hey, this thing can change its sequence at the drop of a hat. This area within your genes is very open to mutation, very, very subject to mutations. And so these mutations cause different types of receptors to be formed. And when you get different types of receptors formed, these different types of receptors are then going to be exposed to antigen presenting cells. And then if the receptor works and it fits the pathogen, then that T cell gets saved. If that receptor doesn't bind to the pathogen, that T cell gets recycled. So in this process, deliberately, when this process occurs at the genetic level, it's very sloppy. Remember when I said that normally when DNA is replicated, you have a 1 in a million frequency for errors. For VDJ recombination and junctional diversity, it's 1 in 100,000, only in this receptor. So you have a 10-fold increase in the chances for mutations to occur in the T cell receptor's DNA compared to all the other DNA in your body. And again, this gives us an advantage from an immunology perspective, because this allows many different variations of the T cell receptor to be generated. What's the variation? The T cell receptor can have as much as 10 to the power of 16 different combinations. This is a 10 with 16 zeros to the end of it. That's the different types of pathogens that your T cells are able to respond to. So if we get back to that original question that I posed about you being with Elon Musk on the surface of Mars and you get infected with a new bacteria, how is your immune system going to respond to it if it's never seen it before? The answer is that because the T cell receptor can basically go through mutations and different combinations over and over and over until it finds the key that fits that lock. And once that occurs and that binding occurs, then that T cell gets saved. And it gets saved in a special place in the body as a memory T cell. So if you come back to Mars in 2 years and you encounter that same bacteria again, you don't have to go through this entire process of trying to find and make a receptor that will fit that pathogen that can eliminate that Martian bacteria. You're already going to have the T cell ready to go. And this is the memory component of the immune system. Any questions on this part? Is this clear as mud for everybody? Okay. And so what we're talking about here with the T cell, the same thing happens with the B cell, with the cell that makes antibodies. And we'll talk about that here in just a moment. But this is a critical part of immunity and understanding how the immune system works. is that we have the ability for our T cells to basically, it tries the key. The key doesn't fit, immediately it can make a new key and try it again. Oh, that didn't work, let me make it again. And keep trying over and over and over and over. And it gets 10 to the power of 16 chances to try to find a fit. So this ability of the receptor to be changed to where it can actually function and recognize a new pathogen is what makes the immune system so powerful, particularly the adaptive part, because it can adapt to a new pathogen and make a new receptor. We've kind of already talked about this process here, but again, this is just kind of reinforcing the positive and negative selection component. If you do end up taking immunology as a standalone course at some point, this is one of the hallmarks of immunity, is understanding this process of positive and negative selection. So if you're somebody that's looking to take immunology here in the next year or so, or maybe you're going to take the MCAT or the GRE or some kind of test to get into graduate school, this would definitely be a slide you want to spend some time familiarizing yourself with, because positive and negative selection is a very important component of immunity and of understanding how T cells work. And again, if the T cell fails either one of these, it's going to be killed via apoptosis and basically recycled again. But this is what allows the T cell to be so tough and so critical in the immune system. It has the ability to recognize a wide array of pathogens, and T cells that are in the position to be able to respond to pathogens have already passed positive and negative selection, and so they're going to be good cells of the immune system that should not do any harm to you, but they're going to kick ass and when they come into encounters and pathogens and do a great job of eliminating those pathogens. Okay, so just kind of summarize the last 3 slides. Your immune system has got a bunch of T cells that can go out and destroy in almost any foreign protein that they encounter, even that Elon Musk Martian bacteria. So we've talked about, again, we're the new pathogen. You've been able to evade the innate immune response. We talked about what's going to happen with the adaptive immune response. You're going to encounter T cells. T cells are going to be able to clear the infection, hopefully. But what happens if a T cell binds to a non-infected cell? So we talked about the analogy of the T cell, the cops coming and knocking on your door. And just as I used in the example with the police, if the T cell knocks on the door, it needs to know that the cell is in trouble. Just opening the door does not tell the T cell very much. The T cell needs to have the door open and someone needs to yell, "Help, I'm in trouble." And we refer to this as co-stimulation. If you think back to the cartoon we saw with the T cell, we not only have the T cell receptor, but we have this accessory molecule. This accessory molecule is the co-stimulating molecule. And that is the molecule, here it is in the cartoon again right here. So here's our T cell receptor. Here's our antigen presenting cell that's presenting some kind of foreign antigen bound with this MHC protein. The T cell receptor comes along and it looks at this and it says, "Man, this looks bad." And then the accessory molecule is able to reach out. If this antigen presenting cell is presenting this and the T cell recognizes this, the T cell will basically extend this co-stimulatory molecule. It's like shaking hands, and then you like the handshake so much you stick out your other arm. When the T cell sticks out that other hand, if it also shakes hands, this activates the T cell, and the T cell then tells the cell, "Hey, this is a bad pathogen. You either need to go kill yourself, or you need to present more of this, and we need to start secreting chemicals so we can tell everybody what's going on." If the T cell sticks out its first hand in the form of the T cell receptor and interacts with the MHC molecule, but there's no co-stimulatory signal, there's no co-stimulatory molecule interaction, then the T cell doesn't do anything. It becomes anergic and it moves on to the next cell. So you've got to have both the antigen and this co-stimulation occur in order for the T cell to become fully activated. So again, if we use the police analogy, the cop knocks on the door, you open the door, and the cop says, "Are you okay?" You opening the door is the T cell receptor binding. You answering the cop's question, "Are you okay? Yes or no?" If you say no, then this is the co-stimulatory signal. The T cell is going to get active. The cop's going to know that you need help. If you say, I'm fine, everything's okay, officer, have a good night, then the T cell is going to basically close the door and become anergic and essentially move on to the next cell. So you've got to have these two signals together. And this is how the immune system controls the ability and the killing power of T cells. If the T cells go unchecked, they could kill every cell in the body that's not in your sperm, your ovaries, or your brain. That's a lot of cells. We need those cells, right? No one wants to have T cells going in and start killing your heart cells. That'd be a really bad thing. The way that the immune system in the body controls this is that the T cell has to get two signals, the receptor signal and the co-stimulatory signal. If those things don't come together, nothing happens. If it's just the receptor alone, no response. If it's just the co-stimulatory molecule alone signaling, there's no effect on the T cell. You got to have both of these to activate the T cell. So what if you are floating around, and we've talked a lot about T cells, and they specifically are going to focus on looking at cells that are infected. But what if you're a pathogen that's not inside of a cell? When we think about pathogens that infect cells, we mostly think about viruses. Viruses have to get inside of a cell in order to be able to replicate. A virus cannot survive. It cannot replicate outside of a cell. It's got to be inside the cell. Many bacteria, however, don't replicate inside of cells. They replicate outside of the cell. So if you are an anthrax bacterium and you're floating around in the body, but you're not inside the cell, how does the immune system going to get rid of you? If you're an extracellular pathogen, what's going to happen to you? This is where B cells come into play. So B cells, remember we talked about this mother cell that comes out of the bone marrow, and it starts to become all these different cells of the immune system. Some of those mother cells will turn into T cells. Some of those mother cells will turn into B cells. B cells are the cells that make antibodies. Has everyone heard of antibodies, I assume? So the B cells are going to make antibodies. And the antibodies are what are going to help protect you against pathogens that are floating around outside of cells, such as bacteria, for example. So the B cells are going to produce antibody, and they use a mechanism very similar to T cells to generate their receptor diversity as well. So we talked about, let me see if I have a cartoon here for it. So we talked about how the T cell goes through this process. Apologize for giving anyone epilepsy there. So we talked about how the T cell uses this VDJ recombination, this junctional diversity mechanism, to change the receptor of the T cell. The B cell does the same thing. It's called the same, it's the same name. It has the same number of different combinations it can generate, and it uses the same mechanism. So the B cell, just like the T cell, can rearrange its receptor, and it can use different rearrangements of receptors until it finds a receptor that will bind to the antigen presenting cell. So just like the T cell encounters an antigen presenting cell and then checks it and sees what kind of pathogen it's infected with, the B cell does the same thing. And so the B cell, just like the T cell, goes through, when it's first made, it goes through some checks to make sure that it can function properly. The first check is, can it make a B cell receptor? Yes or no? If it can, it gets to survive. If it can't make a B cell receptor, then it goes through apoptosis, it gets killed, and it recycles. The second test is, if the B cell can make a receptor, can this B cell receptor recognize some basic antibody molecules, the alpha and beta units of IG? And if it can, so just very similar to the T cell, there's a recognition check. And if the B cell passes this recognition check, then it is allowed to become a functional B cell. It's referred to as an immature B cell. Most cells of the immune system are called immature until they get activated by a pathogen. Then they are called a mature B cell or a mature T cell. but it's a very similar process. Is the B cell able to make a receptor? Yes or no? Is that receptor able to be functional? Yes or no? If these two things are a yes, the B cell gets to survive. If either one of these are a no, the B cell is going to be recycled until it gets the job right. And again, B cells can go through this recombination process just like T cells can to be able to make new receptors. One of the differences, however, is that the B cell receptor is extremely susceptible to mutation compared to even the mutation frequency for T cells. So remember I said for T cell receptors, that mutation frequency is about 1 in 100,000 or so. In B cells, it's 1 in 600. So this pretty much ensures that your B cell receptor, which this is the B cell receptor, it is an antibody. And this diversity process ensures, because there's a high mutation rate in the gene that makes this molecule, that you're going to get lots of different variations. This gives us the ability, again, for us to generate an immune response against crap we've never seen before. So even if you're not going with Elon to Mars, if you're running around somewhere down in South America and you come across a pathogen, a bacteria, or a fungus that your body's never seen before, this is how you're going to get protection, because the genes that can respond against these infections are very susceptible to mutation. And that's a good thing, because the more that these genes can mutate, the more chances you have to generate a new key to open that lock. So if we look at this structure of the antibody, which is also the B cell receptor. So the difference between an antibody and the B cell receptor is that the antibody is, when it's stuck to the B cell, it's called the B cell receptor. When the antibody is not stuck to the B cell, it's just floating around, it's called an antibody. But both molecules, even though they have different names, are the same thing. So here is our antibody, and you've probably all seen this before. We usually use a cartoon image of a Y to demonstrate the antibody. These are the binding regions. We call this the FAB. These are the regions that are going to bind antigen. So there's a lot of genetic diversity and high mutation frequency in these gene segments that encode these proteins, because these are going to actually physically bind to the antigen. So you need to have the ability to make changes here to recognize new things. So we have this binding portion of the molecule up here. And then down here on this stalk, we call this the FC region. And this is the part that's going to bind to cells and also activate a system in the body called the complement system, which is another subcomponent of the immune system that's used to help eliminate pathogens. But the main takeaway from this cartoon that I want y'all to pick up is that the difference between an antibody and the B cell receptor is that the antibody is not stuck on the B cell. It's floating around. The B cell receptor is just an antibody that is stuck to a B cell. So if we kind of go back to this cartoon here, this is considered a B cell receptor because this antibody molecule, see, it's got the same shape. It's got the Y. Here are the heavy and light chains. This is the binding portion, the FAB. Here's the FC portion. But this entire antibody is stuck to this B cell. So we call it the B cell receptor. When this antibody is free floating around out here, it's called an antibody. If we look at antibodies, and we've got 5 major classes of antibodies, we see that they all share a very common structure, which gives it the ability to have a large range of flexibility when it responds to various pathogens. And then if we look at the different types of antibody, this is primarily done, antibodies have been categorized based on where we find them in the body and what their activity is. So for example, we make an antibody called IgE that's used to fight off parasitic worms. We don't have a problem with parasitic worms very much in America, although people sometimes do get infected with them. But in other parts of the world, particularly in places where water is not always clean, drinking water is not always clean, people walk around a lot outside barefoot, you get a small cut or something on your foot, And a parasitic worm can get inside that cut. Very oftentimes, parasitic worms will live in and lay their eggs in little bodies of water. So if you are, just going to make up an example, if you're someone living in a village in India and you're going down to the Ganges every morning to get water and to wash clothes and that kind of stuff, and you're walking down there barefoot and you step on a sharp rock and then go down there by the water's edge, your foot's probably going to be wet. You're probably going to get a parasitic worm infection through that cut on your foot. The skin barrier is broken, so you're now exposed to the chance of getting a parasitic worm infection. We make a class of antibody called IgE that specifically protects you and targets parasitic worms. We make a class of antibody called IgA that's basically found in your tears, your saliva, and in the mucosal areas of your body. What are the mucosal areas? Well, this can be areas like your nares, where you breathe from, your throat, your trachea, your mouth is somewhat considered a mucosal area. The perianal area, where your anus is, that's considered a mucosal area. For women, the vaginal tract is considered a mucosal area. For men, the epididymis and the urinary tract that runs down the sheath of the penis is a mucosal area. All of these mucosal areas, areas where there's mucus, your lungs, all have high levels of IgA to be able to respond to pathogens that you might breathe in or maybe you consume if you eat food. IgG is the class of antibody that's present in the greatest concentration. Most of the antibody floating around in your body right now, if you pulled out all the antibodies in your body and looked at it as a pie chart, about half of the antibodies floating around in your body right now is IgG. And this gives you a broad level of protection against everything. When we look at the kind of protection that pregnant mothers provide to their children through placental immunity, it's going to be IgG. And then we have some other things such as IgD and IgM that also are present. They're not present in as high of levels, but we've got these 5 classes of antibodies to help give us some broad general protection against a variety of pathogens. And just like we talked about with T cells, the antibodies can again undergo these rearrangements to very specifically target certain types of pathogens. So let's say you've been infected with the influenza virus. You have T cells that are going around because it's a virus. They're killing all of the cells that are infected with influenza virus. But some of those influenza viruses have been able to escape infected cells, and they're now floating around in the body looking for a new cell to infect. Antibodies can come and interact and actually physically coat. They'll start to physically coat and cover that influenza virus. And so many antibodies will cover that influenza virus virion that they'll actually make it heavier and more dense than your blood. And the stuff will precipitate out of your blood, which means that when your blood goes through your kidneys for filtration, anything that's heavier than the blood is going to get filtered out. So antibodies play a very important role in being able to help respond to things that are floating around outside of cells that T cells might not be able to kill. In this cartoon up here, we're seeing what an antibody can do against bacteria or maybe a toxin that's being produced. So once these antibodies have been produced by B cells, they have 3 primary roles that they can carry out. The first role is neutralization, which is essentially the antibodies. And again, there are these yellow Y-looking molecules. The toxins are the red dots. The antibody can come and bind up the toxins. And once these toxins are bound, they can't do anything else. They can't go interact with any other cells because they've got antibodies stuck to them and they can't do anything. So they eventually end up getting filtered out of the body. The same thing can happen with bacteria. These antibodies will float around, and if they see a bacteria, they'll start to cover the bacteria. And once the bacteria gets covered with enough antibody, it's either going to get filtered out of the kidneys, or what's more likely going to happen is one of those cells that roam around the body, constantly gobbling everything up that looks bad, is going to come across this bacteria coated with antibody and say, oh, yummy. And it's going to eat the whole thing. And then once it's been ingested, the bacterium is destroyed. This process is referred to as opsonization. And then the third thing that antibodies can do is they can come along if they find bacteria floating around. Maybe some of these bacteria are trying to settle on the surface of your lungs. They've been able to amass there in high enough numbers that they're starting to colonize and settle on the surface of your lungs. Antibodies can come along and bind to the surface of the bacteria. And when they do, there's a system of proteins that are always floating around in our body called the complement protein system. And complement proteins will start to bind to these antibodies that are stuck to the bacteria. And eventually, these complement proteins form holes in the bacteria. And once you poke a hole inside of a cell and you allow stuff that's outside the cell to start to rush in, if you think of the analogy of blowing up a balloon, if you take a balloon and you hook it to a water hose and crank that water hose on, eventually that balloon's going to fill up and pop. The same thing happens with cells. If we can start to poke holes in the cells of bacteria that are floating around in our body, water from outside the cell is going to rush into the cell through the process of diffusion, moving from high concentration to low concentration. Water will rush into the bacteria and the bacteria pops like a balloon. So this is done through the process of complement. But these three mechanisms, neutralization, opsonization, and complement, are all mediated by the production of antibodies. And so just like we saw with T cells, you've got to have antibodies being produced by B cells that can also go around and bind up the pathogens that the T cells can't get to. Any question on the B cells or the T cell stuff? Okay, good. So just to kind of wrap this up here a little bit, as I kind of mentioned, and this is some of this is just kind of my personal perspective, but I think when you look at the different systems that we have discovered so far on the planet from a biological perspective, in terms of complexity and importance, significance, if you will, probably the only thing that's more important than the evolution of DNA as a storage molecule for genetic information has been the immune system. If you don't have an immune system, you're not going to have a living organism, because we're constantly being exposed to pathogens in the environment. Not just us. Animals out there are going through they're fighting the same battles that we are. Many of the pathogens that infect us also infect animals. And so if you don't have a way to defeat pathogens, if you don't have an immune system, that organism's not going to survive. And as I said earlier, we know from doing studies in newborn mice and also looking at women that have had, spontaneous abortions, that if you have severe mutations in your immune system, evolution has decided that that's a lethal mutation, and that it's better that you not be born at all than be born with an immune system that doesn't function. Now, having said that, we have seen some individuals that have developed that didn't have functional immune systems. These are the people that we call like the bubble kids, right? Everyone's probably seen these. These are the little boy, the little girl that ends up living in a sterile bubble their entire life. They can never touch anybody or anything. They can never leave the bubble because they have no immune system. They have no B cells or no T cells. And if they leave that bubble one time and get an infection, they're probably going to die. Sadly, the same thing happens as we get older. As you start to age and get older, the components of your immune system start to wear down. Again, from the body's perspective, you're replicating about half the cells in your body every 24 to 36 hours. Over the course of 80, 90, 100 years, that system's going to start to run out of steam. It's going to start to run out of gas. You may start to accumulate mutations that you didn't have previously. And so your immune system starts to function less the older that you get. And so the immune system plays a critical role. If it's not functional, you're going to have problems moving forward. One thing that we've also noticed is that as the immune system has evolved, the pathogens have evolved along with them. We often refer to this when we look at infectious diseases as eternal biological warfare, because, you can see this and you can generate this in the lab where I've made antibodies. There's techniques you can use to make antibodies. I've made antibodies to viruses and then expose the viruses to those antibodies for 6 to 8 months, and then extract DNA and sequence DNA, and you'll see the virus start to mutate the receptors that it's using. The genes that the virus uses to encode its receptors start to mutate. so the receptor can get away from being bound by the antibody. Well, what's going to happen when that pathogen evolves that new receptor? The antibody, the B cell, is going to start to rearrange its genes through the recombination process we talked about, so it can now recognize the pathogen. So as the immune system interacts with pathogens, there's constant pressure on both the immune system and the pathogen to mutate, to change, because what does the immune system want to do? It wants to kill everything. What does the pathogen want to do? It wants to replicate everywhere. And so many times when we talk about immunity, it's really a battle. It's a numbers game. When we look at people, when they get infected with certain diseases, many times the question is, can we keep this person? And we saw this with the COVID pandemic, right? None of us had ever been infected with COVID before. And that first year, a lot of people died because it was a brand new pathogen. And some people just don't have as good of an immune system as other people. And so many times when people get sick, from a healthcare perspective, the question is, can I keep this person alive long enough for their immune system to recover, to start to respond against the infection? It becomes a bit of a numbers game. The same thing is happening from the pathogen's perspective. The pathogen's goal is to get in and make as many copies of itself as possible, to go from 1 million bacteria to 10 million bacteria, because if it can get those big numbers, it's going to take the immune system a lot longer to clear the infection, and so the pathogen gets to live longer. So there's a constant struggle that's been going on, and we've been able to look at this from DNA sequencing from different samples and different tissues that we've recovered over the last couple thousand years to see that this is something that's been happening for a very long time. The influenza virus that we see every year changes every year. That's why every year there's a new flu vaccine, because 4 months after the vaccine is produced, the influenza virus has changed its makeup, its genetic makeup, and we have to make a new vaccine. So this constant biological warfare requires the immune system to constantly evolve. And that's why you need to have all these pieces working properly together to make sure that you're going to be protected. So this kind of wraps up the lecture here on the immune system. I threw some little questions in here at the end, just to kind of give you some stuff to kind of stimulate and kind of think about. So if you think about smoking and that kind of stuff, and you're thinking about which part of the innate immune response would it impair, we talked a little bit about the VDJ recombination and how having this sloppiness is kind of a benefit for us within the context of generating new responses against pathogens. And then some small questions down here just to kind of think about what's kind of these things would most likely be able to stimulate an adaptive immune response. So I'd be happy to take any questions right now if we've got any. Any questions or comments from the peanut gallery? Everyone's awfully quiet. Let's see if there's a chat. Maya says yes. Okay. No questions. Everyone's good to go on stuff? Okay. Well, we're almost to about 1 o'clock today, so I think we'll go ahead and probably just end a few minutes early today. I don't want to get into these last couple sessions. They can be a bit lengthy. This was probably the longest session of the day. Let's take a look real quick at the other sessions that are coming up and see. Okay, so this one is about, there we go. Okay, this one might be a bit lengthy. Let's see. See what, how long this one is. Okay, so this one's 25. So we'll probably try to shoot for getting through 2 sessions tomorrow, and then we'll finish up with the final session on So we'll knock out 2 sessions tomorrow, the bioinformatics and the regulation of genetic testing. And then on Thursday, we'll knock out DNA sequencing, and then we're going to spend a little bit of time left on Thursday kind of showing y'all some different resources that are available for you to take a look at. NIH has spent a lot of money making some different programs that I'll give y'all not access to, but I'll show you how to find that you can use, where they've actually made like games for looking how DNA mutates, how viruses infect, some different kind of stuff like that. There's actually some virtual science experiments that you can do. You can run DNA on a gel and that kind of stuff. So when we finish up the lectures on Thursday, after we do DNA sequencing, I'll share all those resources with y'all. And then what I'll probably gonna go ahead and do is that, as I said earlier, we're not gonna have the website for the center up until probably sometime after the 1st of June, because we transitioned from the medical school to the College of Science, and so they're having to redo the website. So what we'll do, what my plan is after we finish up the sessions on Thursday, I will email these lectures to everybody that attended the class, and then I'll also email along your certificate as well. So that way you at least have access to the slides. You're free to use them as you see fit. And then you'll have the certificate as well. So I'll send that out on a Thursday or probably Friday after we wrap up the course on Thursday. So tomorrow will be bioinformatics and regulation genetic testing. And then we'll finish up with DNA sequencing on Thursday, share some resources, give y'all some links to some stuff. And then we can kind of move forward from there with the certificate and make sure everyone gets a copy of the slides. That's all that I have for today. If no one has any other questions or comments, then we'll go ahead and wrap up for the day. Natalie, nothing to say?
No, I understood it completely. You teach really good.
Okay, well, hopefully that wasn't too much. And if y'all do have any questions or comments about this, please reach out to me and let me know. And like I said, I'll have to share the slides with you. If you have any other questions after the slides are pushed out or anything, feel free to reach out to me and stuff. But if there's no other questions or comments, then we'll wrap up a little bit early today. I know we went a little bit over tomorrow, and I'll see everyone tomorrow. We went over yesterday, and I'll see everybody tomorrow.
Okay, thank you.
All right, thank you.
Thanks, everybody.
Bye,
See you.
sir. Thank you.
Thank you so much.